All right, so we're going to draw an animal cell, all right? And I'm going to draw it fairly quickly. I'm not going to go into any detail about the functions of the structures, but I just wanted to give you a lay of the land to identify the structures in the cell and to show that this is a great way to practice and study and review the different parts of the cell. All right, and I'll go fairly quickly. So an animal cell, of course, just like all cells, has a plasma membrane. All right, so I'm just going to label that as PM, plasma membrane. Animal cells are eukaryotic cells and they have a nucleus. All right, and the nucleus is a double membrane, so I'm going to draw the two membranes surrounding the nucleus. It's hard to draw them as close as they really are, and that outer part of the membrane is contiguous with the endoplasmic reticulum, which I'm going to draw like this. All right, and it's again much more extensive than this in cells, and it depends on the cell type. It looks a bit like an octopus, doesn't it? But anyway. Okay, within the nucleus we've got a dark region of the nucleus, but it's not enclosed in a membrane, and that would be the nucleolus. And then the nuclear envelope is studded all over, and I'm going to draw these much bigger than what they really are, with these pores and pore proteins, and I'm going to draw the pore proteins in red. And so these pores allows the movement of substances into and out of the nucleus. All right, there we go. So let's label some of these then, some of these structures. All right, so here we've got, of course, the nucleus, and I'm just going to write the letter N. Here we've got the nucleolus, I'm going to write the letter N again. And here we've got a pore, and I'm just going to write NP for nuclear pore. All right, so the nucleus, of course, contains the DNA, and there's lots of traffic into and out of the nucleus, but the DNA is nicely protected inside the nucleus, and things can move in and out through these pores. The nucleolus is this darker region inside the nucleus, and it's where ribosomal RNA is made. All right, so now let's look at the endoplasmic reticulum. The endoplasmic reticulum has got two major regions to it, and one of them being the rough ER, and it's called the rough ER because it has ribosomes that are embedded in the membrane of the rough ER part. And I'm not going to draw ribosomes over the entire rough ER. I guess maybe I will. It's pretty easy to do. All right, there we go. So that's the rough ER. Now these parts of the endoplasmic reticulum that have no ribosomes, they're called the smooth ER. Now ribosomes can either be embedded into the rough ER, or they can be free, sort of floating in the cytoplasm there. All right. Okay, so let's label some of these structures then. All right, here we've got the smooth ER, and that's because there are no ribosomes embedded. And then we've got this rough ER region, RER. Okay, and that's because it has ribosomes embedded on it. And so we've got two different kinds of ribosomes. We've got our bound ribosomes, and we've got our free ribosomes. So ribosomes are the site of protein synthesis. It's where amino acids are joined together to produce proteins. And the rough and smooth ER have a variety of different functions that I've gone over in lecture. All right, so now let's add the Golgi apparatus. So the Golgi apparatus is kind of this system of stacked, flattened sacs. All right, and it's where products made on the ER are transported to very often, and it's where they're modified or stored, and of course they're transported in little vesicles. All right, so each one of these is membrane enclosed, little vesicles, and they deliver their contents to the Golgi apparatus, and then they kind of move through the Golgi apparatus in vesicles, and then they can emerge from the other side, modified or altered and packaged. So there's kind of directionality, all right? Things can move through the Golgi apparatus, and they kind of move in that direction. So this is the cis face of the Golgi apparatus, and that's where things are received, and this is the trans face where things are shipped from the Golgi apparatus. So let's add some labels then. So here we've got the Golgi Apparatus, and I'm going to go GA. And here we've got vesicles. All right, I'm just going to label them as a V. All right. So next I want to add some larger specialized vesicles known as anybody? All right, let's do a couple of big ones there, known as L4 lysosomes. All right, and these are specialized vesicles, and they contain lots of highly destructive hydrolytic enzymes, carbohydrase enzymes, nuclease enzymes, lipase enzymes, protease enzymes, and of course what they're designed to do is to break down all of those macromolecules. All right, so let's see, what do we got next? I'm going to add a mitochondria. All right, I'm going to add one over here. Now cells vary in the number of mitochondria that they have. Some cells have very few and some cells have very many, like cardiac muscles might have 500 or so mitochondria. And it's a double membrane structure. We've got an outer membrane and an inner membrane, and this is the site of many of the steps, or most of the steps, of cellular respiration. All right, so there's M for mitochondria. All right, so that's most of the major components of a cell. I'm going to add a few more little details to it, and I'm going to add those in green. So on the outside of the plasma membrane, I'm going to kind of do it as these X's and squiggles. We've got what's outside the cell membrane? Extracellular matrix. All right. And then running throughout the cell, and I can't draw it as extensive as what it really is, we've got different components of the cytoskeleton. Now remember, there are 3 different components of the cytoskeleton. We've got microtubules, microfilaments, and microtubules, microfilaments, and what's the other one? Well, let's draw them, see if you can remember that. And I'm just going to draw it as a system of sort of cables running throughout, and it really does extend throughout the entire cell and throughout the cytoplasm. All right, and I think the last thing I need to label is the C4 cytoplasm. All right, so they're all the major components of the cell. All right, I think that's it. See if you can do it.